Okay.
So you can start.
It's not showing my display, right?
It will take time.
Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and this is our third and the last live session for the course Genetic Engineering Theory and Applications. So if you have any queries, you can post your questions and I'll be happy to answer.
I have a few questions on the sheet.
So questions, I think we discussed last in the last live session also. Question is going to be of multiple choice questions. So it's going to be objective questions. You're going to have 100 questions and each question is going to be of 1 mark. There will be no negative marking and there will be no long answer or the long answer type of questions. So it's going to be objective questions, 100 questions you will get in the exam and for 100 marks, and there will be no negative marking. Uh, Shashvi, I think you should ask these questions to the NPTEL office, whether they will give you the assignment marks before the exam or not. In my opinion, probably not. So, but it's better to clarify from them because I am only conducting the course, but as far as all the technical issues or operational things, it's better to contact the operational team back in IIT Madras, and they'll be happy to help you. Abhijit is asking, can we use cancer to our benefit to cure neurodegenerative disorders? Now, if you understand the cancer properly, it's not about the growth. It's also a growth of the cells without any function. Okay. Otherwise, So you can imagine that if somebody is getting a liver cancer, the liver cells are actually growing. That does not mean that this guy is doing better functions in terms of liver functions, right? Its liver is going to destroy while these cells are growing. So the cancer cells are having a proliferative potentials, but these cells are of no use. So in case if you induce the cancer, the person is anyway having a neurodegenerative problems. On the other hand, whatever the remaining nervous system he has, that is also not going to be functional, because you are going to take up the nutrition from the healthy tissues, and the cancer cell is very good in terms of driving the nutrition from the healthy tissues, and that's how the person is going to be even going to show more pathophysiology of the neural pathology and those kind of issues. So cancer cannot be used as a uh, option to cure the neurodegenerative options, because it has a proliferative options. What we can do also is we can use the cancer cells to produce some secondary messengers or secondary molecules, and that can be pure and then supply to these patients as a supplement, and that supplement probably could help in reviving the neurodegenerative tissues. Because what we can do is we can provide some signal to the tissue which is uh, going for the degenerative problems, and then during this, if you provide the right stimulus, it could be possible that the cells may start proliferating. So Vida, I think we already discussed about the pattern. It's going to be a multiple choice pattern. You're going to have the one questions, and you're going to have the four options. Out of these four options, you're going to have one of the options correct, and you're going to have the 100 questions for the exam with the 100 marks, and you can attempt as many questions you like. There will be no negative marking. So Divya is asking more about how we can prepare for this exam. So you should prepare for the exam if you go through with the content, especially some of the important questions. The other issue is you should also go through some of the assignments questions which are being wrong for you. For example, you can just very quickly go through these assignment questions and see which question could not be able to do that, and then you can practice for that particular questions. And if you go through by the assignments, it is very quickly you will be able to revise the whole course, and you can be able to attempt a much better way than if you try to go with the video lectures or the transcripts. If you have any more questions, you should ask, because this session is very small and I request you that you should take the full advantage and of this opportunity to clear your doubts so that you will be able to do better in exams. No, I cannot give you the details of whether there will be numerical type of questions or whether it will be a subjective type of questions or whether there will be, experimental questions and all that. That is, I think we have to keep some amount of secrecy in that. So I will not going to give you the detail about whether there will be numerical type of questions. But what I can say is that the questions are going to follow the same pattern what we have done so far into the assignments. So in the exam also probably you will see some of the questions which are directly from the assignment. So there will be those questions will be easy to answer. That's why I'm very seriously telling you that you should see all the questions what we have asked in the assignments and try to see those questions where you are wrong and you should attempt those questions again and try to practice for those questions because at least some questions you will get from the assignment as well. uh, I think Yashasvi is again asking how what is the percentage of the questions we are going to ask from assignment and all that. See, these are the very difficult things to tell you, but there is a significant portion of the questions from the previously asked assignments. It could be may not be direct questions, but it could be some question which is related to that. Maybe we might have changed the values or we might have changed the concepts. So if you go through with all the assignment questions, you will definitely going to get some questions and you will get be able to attempt those questions much easily and it will help you in to prepare for your exam. Sai, I think we can. These courses are going to be floated every year. I cannot guarantee you, but most likely it is going to be floated next year as well. But while you are thinking about the next year, you should attempt this year itself and clear the course. if you Soumya is asking about how many assignments, minimum assignments should submitted to pass the exam. So I think if you go through with the instructions, it says that the 8 assignments out of 12 is what we need. So in those 8 assignments, you should get the passing marks. So 8 assignment is the minimum what you should be get attempted. See, most what Abhijit is asking and discussing is very much uh, different than what we have discussed in this course. But even then, uh, see, you don't have to see couple of genes, because what happen is that even if you have a single gene, the single gene is behaving differently if it is expressing at 1 milligram per ml, or if it is expressing at 10 milligram per ml. So even neurodegenerative disease is also showing upregulation of some disease genes. Cancer cells are also showing a upregulation of some genes, but you don't know what are the different counterparts and adapter proteins are actually making a complex, and how the signaling events are being different from the two different tissues. So that's why in one case it is actually inducing the proliferations, in another case it is actually inducing the degenerations. So or the damage of the particular tissue. So because these are the very difficult and fine-tuned controlled events, and so far what we have understood is that even very subtle changes in the expression could actually reverse the function of these genes. So as I said, if you have a even if you have a upregulation compared to the control sample, 1 milligram versus 10 milligram probably could make a big difference in terms of their functions. So we cannot conclude so simply that those couple of genes are upregulating or downregulating, and the pattern is same in both the tissue type, so they should behave similarly. If you have any more questions, you should ask, because this is a very limited session and I request you all to take the full opportunity. So far, I think students are more worried about the exam pattern and question pattern and all that, which is we can discuss also. But apart from that, I think if you have any confusion of about any content or any aspect what you could not understand, you should take the benefit of these live sessions. Otherwise, we'll wind up very soon. So Vignesh is asking about the examples of the resuspension buffers. Resuspension buffers could be any buffer where your cells or where your bacteria or where your host cell is stable, or the resuspension buffer could be a buffer where your product of your interest is stable. So you can be, resuspension buffer could be as simple as like simple saline, where the like osmolar saline, where it can be just all your cellular content is going to be intact, or it could be PBS, it could be tris, it could be something. So there's no hard and fast rule about the suspension buffer. The only criteria is that the suspension buffer should be able to keep the integrity as well as the quality of the product what you are trying to isolate from the cell. Ruchira, I think we already discussed. All the questions are going to be of MCQ type, no essay questions, no negative marking, no subjective questions. If you have any more questions, you can drop your questions. Otherwise, we'll wind up this session in another 1 minute. So Deepa is asking something which is more technical actually and related to exam. I request you to contact the IIT Madras team which is going to do all this and will give you the passing marks and maximum marks and all that. So Soumya is also asking something which is more of a related to exam only. So I think these questions, how many weeks should we cover in minimum? I think we have to cover all the 12 weeks because we are going to have the questions from the all the assignments, all the weeks. Content is going to be 100%. So I cannot tell you which content I have elaborated or which content I have focused while I am preparing the questions. If you have any more questions, you can ask me. We have very small time left now. Sir, what all we have to do in PhD? Um, there is no need to do PhD. The idea is that you should understand the course very nicely. That may help you to prepare for different types of competitive exams. Once you clear these competitive exams and you have the interest to pursue a scientific problem, you can pursue a PhD program. If you have the if you want to pursue a product development and something related to industry, then you go to the industry jobs. And some of these majority of the competitive exams actually allows you to do that. So for example, if you clear the gate, you have multiple avenues. You can go to the companies, you can go to the PSUs, you can go to the any IIT for doing the MTech, or you can go to the IITs to do the PhD. So once you, I think, go through with the subject, make it very, you know, prepare it well, and then you clear the competitive exams. And depending on your performance in the competitive exams, your scores and other things, you can take the decision where, what you want to do in future. You can do a couple of pull down essays, Vijit, if you want to do such kind of experiments and you want to solve the questions. So if you pull down, I think you will be able to get all the factors which are sticking to each other and down regulating the or delineating the downstream signalings. And if you do so those kind of experiments, you can even do those experiments organelle wise and membrane and cytosol and those kind of. So there are multiple compartments through which you can actually do these kind of experiments. So if you have any more question, ask me so that you can take the full advantage of this session. Okay, so if you don't have any more questions, I think we'll wind up this session. Uh, I hope you have liked this course, and I hope we could have been be a helpful resource person. And so all the best for your exams. And when you prepare for your exams, you should consider preparing for the questions what we have asked in the assignments. And on the other hand, you should also go through the content. We have provided you the transcripts. So transcripts are actually going to helpful in terms of going through the content in a much faster way. Don't just be do the shortcuts, because I have asked the questions from the every week assignments. So it is very difficult to even clear this exam or clear this incoming exam simply by going through with the limited content. So with this, I would like to conclude our live session here, and I would wish you best of luck for your exams. And if you have any more queries and any more questions and confusions, you can directly get back to me even after the course also. Thank you.